also Professor Peter Hagar, and also other participants. So today we would like to hear the wonderful lecture from Professor Rosalind Anderson from the Wisconsin Medicines regarding the title of the title is the calorie restriction and nutritional modulation of aging. So maybe before we start the lecture, so is it fine if we would like to invite the Professor Badrul Hagar for giving some brief opening statement. So Professor Badrul Hagar, so the time is yours.
Okay, baik. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Good evening. Our Honorable Professor Rosalind Anderson, PhD, as our guest speaker. Also, Dr. Agian J. Villano Barida, PhD, as our host and also our moderator this evening. And of course, all the participants. Welcome to webinar Innovation in Aging Science as part of webinar series of Metabolic Disorder, Cardiovascular and Aging Research Cluster in Mary FKUI. I think calorie restriction being a big interesting issue right now is widely used to study aging processes. Some experts believe it is a simple and highly reproducible method for delaying the aging process, preventing the onset of aging-related disease, and also extending average or maximum lifespan. People who reduce the calorie they consume has found that such restriction can slow down human metabolism. However, there are still many points that cannot yet be answered clearly, which are still ongoing process in research or become challenges for convergent research in the future. For example, some questions: why should calorie intake be decreased as a person grows older? Or what are the role of calorie restriction and diet quality in promoting healthy longevity? And also, how does calorie intake affect aging? I think there's some of these questions will be explained by our guest speaker, and some of them still become a research question for convergent research in scope of molecular and cellular biology of aging. I think it is amazing opportunity for us to have you, Professor Rosalind Anderson, to discuss such challenging issue. Of course, I hope the collaboration between Emory FKUI and the University of Wisconsin Medicine USA in metabolism of aging program will not only in scope of knowledge transfer in scientific meeting, but also in clinical experience, including research. Thank you once again very much, Professor Leslie, to become part of this event. And also to all participants, please enjoy the meeting. Take care all. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih.
Okay, so before that, so is it fine for us to get a picture or photo together with Professor Anderson? Is it fine? Okay, so please turn on your video so we can make a photo together with Professor Anderson and also Professor Badrul Hagar. So please maybe
stop sharing my screen. Let me take that away so we can see the whole gallery. Look at that.
Sure.
Wow.
Thank you. Fantastic. So maybe Mbak Tika or
yeah, baik Dok.
Yeah.
Okay. Let's get I get screenshot. Is first slide 1, 2, 3, and the next slide 1, 2, 3. Okay.
That's fun. Okay, I'll go back to my slides. Share. There we go. Okay, so will I kick off? I can't hear you.
Okay, so now we will hear the wonderful lecture from Professor Rosalind Anderson. For the title of the lecture is the calorie restriction and nutritional modulation of aging. Okay, so the time is yours, Professor Anderson. Thank you very much.
Super duper. I'm so thrilled to be here. Thank you for the invitation, Jeffy, and thank you all for coming at this hour of the evening. It's I'm still wrapping my head around the idea that we're other sides of the planet. I'll keep things nice and simple. I know we have a limited time, and I'm cutting into your evening time where you probably should be home with family. or in the lab doing research. And so I'll just give you some broad strokes of the research that we've been doing. So I study caloric restriction as a means to understand aging. And we've known for 100 years now that a lower intake of calories over the lifetime of an animal, while avoiding malnutrition, can improve survival and prolongs health. And this is true for multiple species that have been tested over the years, with most of the work done in rodents, as in laboratory rats and laboratory mice. We embarked on a study in non-human primates. These were rhesus macaques, born and raised in captivity, which is sad for them, but good for us. And we were able to show that even in a long-lived primate species, we see the changes in the onset and progression of numerous age-related diseases. So these circles here are from data from our monkey study. We started with adult animals and put them on a caloric restriction diet and then followed them until their natural death. And you can see all of these different diseases and disorders: cognition, sarcopenia, brain atrophy, incidence and progression of cancer, diabetes, and of course of interest to you guys, cardiovascular disease. So it seems extraordinary that all of these diseases that are on the surface unrelated to each other, they occur in different organs and tissue systems, and they even have different institutes and foundations to research them, and yet they have this shared component, which is an increased risk as a function of age. And it turns out that CR can actually impinge on each of these. And the reason it's doing that is because it's changing how aging happens. So we have a couple of key questions. What creates the shared vulnerability to this collection of unrelated diseases and disorders? And what does CR do differently that might tell us what's happening during aging to create that vulnerability in the first place? So myself and many of my colleagues have a suite of things that we look at when we're thinking about the mechanisms of CR. Our goal is to understand what it's doing, so we might have a better idea of what's going wrong, or at least creating vulnerability during aging. And so these are the same old soldiers you might have seen 10 or 20 years ago. And the reason that you keep seeing them is that these were discovered in very nice experiments in things like yeast and worms. And that fundamental biology has held true. And as we go into more complex organisms, mammalian systems, we still find these same players in regulation of longevity, but also in the mechanisms of CR. So I'm showing a few of them here. I don't know if you can see my mouse. Can you see my little arrow? NAD, these are the metabolic indicators. So this will be of interest to all of you guys working in metabolism, whether it's diabetes or cardiovascular disease. We have these metabolic indicators that are frequently the targets of drugs and certainly things we look for in terms of biomarkers. Then we have these regulators that come in and detect changes in those factors. So for example, sirtuins are sensitive to NAD, AMPK is sensitive to the levels of energetic currency, adenosine monophosphate here, although it's the adenosine triphosphate that's our energy source. Insulin IGF signaling is conferred through this important kinase AKT, you'll have seen, which is actually also one of the first oncogenes discovered. mTOR, super popular at the moment. A lot of people are interested because of the longevity effects of rapamycin. And then PPARs, these are peroxisome proliferator activated receptors. Maybe some of you in the cardiovascular realm will have come across these, certainly in diabetes, the TZDs as they're called, and they sense fatty acids. And then occluded by my picture of all of you guys, P53, which is another oncogene. So we know that these factors are important in caloric restriction. Each of them independently has been implicated in longevity regulation through genetic studies. And what they do is that they sense metabolites, energy, oxidation sensors, and all of those are working together to implement CR. And so at the cellular level, because often we look at whole organism and then we collapse down to molecules, and it's difficult. We're still missing that bit where all the cells communicate as a tissue, but we're getting there with systems biology and single nucleus sequencing. But within the cell, you can see where some of these factors connect and how they signal to change the behavior of cells. And of course, I will tell you, I'm very biased. To my mind, metabolism is the be all and end all and most important thing that we should be looking at because it connects to disease vulnerability. But you can see whether we look at PPAR agonists, NAD precursors, resveratrol, metformin, one of the most popular diabetes drugs, adipiron, a fairly new drug that came out of Japan about 10 years ago that activates adiponectin receptors, or rapamycin. You can see this constellation of cellular functions that are all going to be responsive to these drugs: mitochondria, protein synthesis, autophagy, Golgi function in the secretory pathway. And of course, well, my favorite here is PGC1 alpha, but I'm not going to talk too much about that today. So you can see it's very complicated. We've got a lot of signaling factors, a lot of effector molecules, and a lot of cellular processes that are responding to things that we know regulate aging. The question is, how do they all fit together? So we did a study a few years ago where we took tissues from a whole bunch of different mice that had been on caloric restriction. And I'm showing you here brain regions: cortex, hippocampus, heart, liver, kidney, white adipose tissue, and skeletal muscle. This is gastrocnemius. And instead of asking what genes individually are going up and down, we went straight and asked what pathways are changing. And what we were hoping to see is some kind of coordination among tissues in response to CR. And that's what we saw. In fact, we could see this pathway, this signature of mitochondrial activation and immune function downregulation or depression of immunity. Now, by that, I mean that the inflammatory and immune tone was down, not that the system was broken, just that it wasn't hyperactivated as you find in aging. So this signature of increased mitochondrial function, not necessarily number, and downregulation of inflammatory pathways was shared across these completely different tissues. So we had done a couple of different experiments. The PMCID shows you how you could find this paper if you want on PubMed or any other search mechanism. And so we decided, well, if mitochondria are so important that this is happening in every tissue that we've tested with CR, what happens to cells when we change mitochondria? And this is something we published just a few years ago. We took these cells and we overexpressed PGC1 alpha. I pointed that one to you in the scheme earlier. And this is a means to change mitochondrial function. It's a transcriptional co-activator that regulates mitochondrial genes that are expressed in the nucleus. It also regulates expression of transcription factors that regulate mitochondrial gene expression. And so when you change PGC1 alpha, you change the quality, not necessarily the quantity of mitochondria. And this is what we did. We activated them and they became more oxidative. They respire more. They use more lipid fuel source, and they become more proficient at metabolic flexibility in terms of what kind of different fuels they're able to use to derive energy. And we came across this very interesting thing when we started to look at here, either protein abundance or RNA, looking at regulation by post-translational modification. So these are proteins that have, a phosphorylation or acetylation that changes their function. So this is regulation at the protein level, not at the gene expression level, or regulation at the RNA level. So this isn't how much of a transcript is produced, it's how that transcript is handled. So we looked across these different regulatory mechanisms and we saw this profound growth signature. So when you change the mitochondria just a little bit, to dial them up just a little bit, all of these cellular factors involved in growth respond. And they respond not just at the protein level, but at the RNA level. And not just in terms of abundance, but also in terms of other regulatory mechanisms like post-translational modification to change the function or maybe location of those proteins, and at the regulation of the transcript processing. So what kind of proteins are going to be produced by those transcripts, and in a sneaky move by RNA molecules, sometimes those RNA molecules are not converted to proteins at all. They stay there and regulate other RNA processes. So it becomes quite complicated, but you can see a small change in mitochondria precipitates a very large effect. And when we look at RNA-seq data from these cells where we've stimulated the mitochondria just a little bit, along the lines of what we see with CR, in fact, we see broad scale changes in multiple cellular functions: metabolism, immune function, growth, structure, and features associated with cellular homeostasis. So this is telling us it's not a coincidence when we looked in those mouse tissues and saw mitochondria as the shared response across all of CR, because you can change mitochondria just a little bit and have enormous changes across the cell and therefore across the tissue. So I'll switch gears and talk a little bit about the monkeys, because I think this is usually why people are, I only ever get asked to come and visit people because of the monkeys. So the monkeys are Very interesting for us. We don't have wild monkeys. They're not roaming the streets and they don't steal from barrows. In fact, they're only in primate centers and vivariums. And so the ones we have were born and raised in Wisconsin. And in adulthood, they were divided into two groups, control versus restricted. And I'm showing you two monkeys. These are males from the program. And age 27 is the median age of lifespan in monkeys in captivity, by which I mean half of them will die by the time they're 27 years of age. So this is an advanced age for a monkey. You can see very clear differences between the control and the restricted monkey here. It's very clear. So we wanted to get into seeing, well, what's the effect on lifespan and aging? And this work is published. You can see this is the PMC ID here. If you go to PubMed search, we can see, including males and females, both together, we can see very clear separation from control in blue and the CR animals in red. This is age-related mortality as defined on necropsy with a pathologist. This is all-cause mortality, so includes even accidents or unexpected deaths. And then here we have morbidity, age-related conditions. And again, a very nice separation between the control and the CR animals. And then individual diseases, cardiovascular disease, insulin resistance, and cancer, all lower in the CR animals. So we started to get into this idea of looking at the molecular signatures of these monkeys, and we were very lucky. We had liver biopsies taken from monkeys at the very beginning of the study. So for these animals, it was in 1989, and then, or actually this was in 1992, and then 2 years later. So they had just about become adjusted to the caloric restriction diet that they would be on for the next, 30 years. But this was the very early stages, and we had liver biopsies. So we went in and we looked at the transcriptional profile, post-transcriptional regulation, this is RNA processing, protein levels through mass spec, proteomics, and also the acetylome because of our interest in lipid signatures and lipolysis as a means to derive energy. I'll show you only the cliff notes. This is published, you can see it right here. This is the PMC. So this is the RNA-seq data, and you can see, just as you might have expected if you'd spent years looking at mouse data as I had, the same signature shows up. We see lots of changes in metabolism. Here, peroxisomes for lipid handling, fatty acid degradation, branch chain amino acids, oxidative phosphorylation. They're our friends, the mitochondria, front and center. And we saw the TCA, also resident in the mitochondria. And we saw the signature of RNA transport and spliceosome regulation, which was new. This hadn't been identified in CR before. Down-regulated pathways include what you might expect, immune pathways and inflammatory pathways. Now, I happen to think that we see this signature because when you change metabolism, you down-regulate growth. And so many of the processes in immune and inflammatory function require either clonal expansion, which is growth, or the creation of new molecules and synthetic processes, also growth. And I think that this yin-yang between metabolism and growth is really at the heart of what's happening with CR. And then you can see the heat maps here showing you the various pathways and the striking differences even within 2 years between control and restricted animals. The RNA processing was particularly interesting because it was new. And what I'm showing you here, you can see in the control animals, very little difference in terms of how their transcripts are being handled and processed. But here we can see, here we can see massive induction of this mechanism by caloric restriction. And the same pathways are being regulated here. So it's RNA processing, peroxisome, branch chain amino acids, but how that regulation is happening is different. What I showed you before was transcript abundance, how much of those transcripts are there. What I'm showing you now is a different suite of factors in the same pathways, but being regulated in the way their transcripts are being processed. So two different mechanisms in the same pathways. Well, it turns out we had transcriptomics, we had proteomics, we had metabolomics, we had lipidomics, we had lots of different molecular profilings put together. And so we actually did just throw them all together. What I'm showing you here are 20,000 molecules regressed against each other, looking for that difference over a 2-year time period. And the striking, I'm not going to go into detail here, but the striking feature, you can see these boxes, right? We called them mega clusters, but they were groups of molecules that appeared to be regulated together. So this is an active response. This is a coordinated response over the 2-year period from not being on a CR diet to being on a CR diet. And that those regulatory, these megaclusters or modules, you might call them, involve different types of mechanisms to exert their effects. So this is the idea that as a function of this prolonged exposure to caloric restriction, there is a reprogramming that happens in the liver to change how metabolism functions and change those growth regulatory pathways as well. We did some machine learning, which was super interesting. This is a very different way of handling the data. Instead of asking what's up and what's down, now you ask, can we arrange the data in such a way that it will tell the difference between control and CR? But you might well tell the difference between diabetes or not diabetes, or cardiovascular disease and not cardiovascular disease. So you put in your molecules and it ranks them, it creates these lists, and then it will reach multiple iterations of going through the data and building what's called these trees, which is why it's a random forest, and you'll be able to distinguish between one disorder and the other. And we do this, and interestingly, it brings out the same pathways that we'd identified before, but it gets them using a different method. And so we see immune and inflammation, fatty acid metabolism, and carbon and sugar metabolism. And it was super interesting because these pathways intersected when we put together the gene model, the protein model, the acetyl protein model. They come together and form this beautiful network, which we think is the signature of CR. So these are the types of approaches we can use to try and understand the mechanics of what's happening under the hood, so as to speak, with caloric restriction. What is it doing to change the function of tissues? So this is one of the hallmarks of CR in monkeys and mice, a reduction in body weight, a reduction in fat mass in particular, improvements in circulating glucose levels, which are lower. So what I'm showing you here, these are the controls, these are the CRs, this is time point 0, this is 2 years after being on the diet. So for example, here I'll show you the glucose. The control animals don't see a change in their circulating glucose. The CR animals have a significant reduction in their circulating glucose. The control animals, no change. You can see a spread in the insulin values. The CR animals snap down to a much lower fasting insulin, and then the insulin sensitivity. So these hallmarks that we see in monkeys are just like we would see in mice, but they are also just like what we see in people. So these are data from a human clinical trial of caloric restriction that was conducted over 2 years by colleagues of mine at different locations across the United States. And in humans with 2 years of CR, you can see lower body weight, lower adiposity, glucose is lower, circulating, fasting glucose, fasting insulin, improved insulin sensitivity, and lower risk factors for cardiovascular disease. It's exactly parallel between the monkeys and the humans, which gives us great encouragement, because it's more likely now that the molecular underpinnings of CR that we see will translate also to humans, which, let's face it, are just another primate species. So I'm going to go very fast now and just touch on a few little things here and there that may be of interest to some of you. We've done some analysis looking at adiposity, because adipose tissue reduction is one of the signature events of caloric restriction and weight loss. For example, if you're in the diabetes or cardiovascular field. And what we found was when we looked at either subcutaneous adipose or visceral adipose, both respond to CR, but the details of the response are not the same. At the molecular level, there are different pathways being introduced or repressed, depending on whether you're in a subcutaneous depot or the visceral depot. And this is super interesting, because we know for a long time people thought adipose tissue was just a place to store fat. We now know that it's an endocrine organ. Then it became clear that there were, inflammatory signatures and things like visceral fat. We now know that different adipose depots behave differently. They serve different purposes, and the response to CR is distinct. But what we were encouraged to see is that in both depots, this RNA processing signature was evident. So we changes in metabolism and growth pathways that are specific to the adipose depots, but this shared feature resides with this mechanistic component of transcript regulation. This is just showing you a little bit more detail here. I'm showing you RNA-seq data. So these are all the transcripts in 2 different depots, subcutaneous and visceral, and the change that happens between control and restricted groups. And again, you can see there's this lots of different transcripts that are being regulated at the level of transcript processing now, not necessarily abundance. And we can see these changes in exon usage. But what's interesting here, this is a tool that's being used. So this is where you either include or exclude different exons as you're processing the transcripts. This mechanism is used by the subcutaneous depots, not the visceral. So this is a very different behavior of the two different depots in response to CR. And these are the signature pathways we see that regulate extracellular matrix, factors related to immune function like migration and complement, and peroxisomes, which you were delighted to see there, because really I'm a bit of a lipid fan myself. We also see changes in skeletal muscle aging. This is just the cliff notes, so to speak. I don't know if you have that term. This is a small book that people use to cheat. Well, it's not cheating, but to reduce the amount of work they do. Instead of reading the novel, they read the short version and then go take their test and hope they've covered all of it. But those short versions are called cliff notes. So just to give you the short version, you can see here the control animals are in orange, and you can see this is leg, this is muscle mass as a function of age. This is years and decades, 10, 15, 20 years of age, so long time study. And you can see this bulking muscle bulks up until about age 16, 17 years of age, and then they start to lose it. And it's quite a considerable decline. So this loss of muscle mass and function is called sarcopenia. What happens in the CR animals, they don't get as big because they're not as big, the animals are not as big, but they retain their muscle for longer, and then they see a more gradual decline. And so we were able to connect those differences in the trajectory of age to metabolic cost of movement, which I'm showing you here, where there's a very clear relationship between the metabolic cost of movement and retention of peak muscle mass for the control animals, and it's just all gone sideways for the old animals. That connection, that regulatory connection is gone. But we can also show that the difference between basal insulin and retention of muscle mass is there. And so the more insulin sensitive the animals are, the better they are in retaining that peak muscle mass. So there's a connection to delaying aging and maintenance of peripheral metabolic features related to glucoregulation. This is very new, so there's no number associated with this because it's not published yet. And in fact, the adipose story isn't published either, but Tim Rhodes is the guy who put this together. And we can see very clear sex dimorphism. So in our prior studies, we'd only looked over the course of aging in the males. Now we can look at the males and the females, and we see big differences. So both of them are protected against sarcopenia. It's much more obvious in males because males have more sarcopenia than the females. But the way in which aging is taking place is not the same in males and females. So we think this is going to be super interesting as we move forward. And I think it's going to have profound clinical relevance, because somehow we still think that females are just small males, and they're not. In multiple systems, as we look now, we can see it's abundantly clear that some of the metabolic underpinnings, the molecular details of aging are not equivalent between males and females. And so we need to look at those differently, because how we treat aging disorders is going to be different. This is my last little piece, and I'm looking at brain aging. This part is published. We were able to show a preservation of gray matter volume. Other papers showed preservation of white matter volume. In the brains. These are the same monkeys now. These are their actual brains in the same monkeys. This was published a while ago. And then key areas that were preserved by caloric restriction, the caudate, putamen. These are areas that are super sensitive to aging, by the way, and the temporal cortex. It was very nice work of a postdoc who was on our aging training program, which I've now inherited as well. Had looked at how those changes in gray matter volume relate to peripheral glucoregulatory function. And he was able to establish that there is a relationship between that age-related gray matter volume loss and the preservation of gray matter volume with caloric restriction. So here are two examples of metabolism in the periphery connecting to the pace of aging measured in individual tissues. We've since gone on to do some mouse studies looking at molecular profiles, and we can see these clear differences as a function of age. And I'm really just showing you that what's interesting here is you get a lot of features that look like innate immunity, so things like viral detection and bacterial infection. We also see inflammatory signals, and we see growth signals. So there are all these changes that are taking place as a function of age, but what's of interest to us is this immune and inflammatory pathways, because people hadn't really considered how immune and inflammation were playing a role in brain. With the blood-brain barrier, became difficult to understand. But we know now, of course, there are resident microglia with our T cells in the brain. There's a lot of crosstalk between the cerebrovascular fluid and the brain. We have the G-lymph and all these other features. So now with and we're just about to embark on some single nucleus sequencing in these monkey brains, but these are the signatures that we see in the mice. So a lot of immune activation and inflammatory activation, a lot of the usual suspects showing up there simply as a function of age. So the upshot is we know that age is changing metabolism. We know that it's changing immune and inflammatory pathways. And I'm not showing you the data now, but we also know that CR changes how that happens. And it's changing the relationship or it's leveraging that relationship between growth and metabolism. So a couple of insights. I hope I haven't gone too far over in time. I'm trying to keep it nice and tight. We think biology of aging research, I think, is a key way to try and understand what's happening with age-related disease vulnerability. I think I've shown you the value of caloric restriction there. Mitochondrial changes we think are very important in the CR program. I showed you the mouse data and I showed you the cell culture data that indicates if we simply change how mitochondria behave, we can change a host of cellular functions that will relate directly to things like immune and inflammatory function. Growth and metabolism are interwoven, and that there are different regulatory processes that are brought in to make those changes happen with CR. And we think those same regulatory processes are influencing the drift that we see with age. And then I showed you this little cliff notes data on skeletal muscle and brain aging that we've connected also to peripheral metabolism. This is my team that work with me in Madison, Wisconsin. I'm so lucky to have a very talented group of individuals, and we've got representations from all over the world, which is lovely. I love to have an international flair in the lab. It makes for great science. And then these are the funders and former lab members and my collaborators, in particular for funding from NIH, NIA, and some foundations like the Simons Foundation and Impetus grants that were more recently acquired. So with that, I can stop sharing, and I'd be very happy to answer any questions you might have.
Okay, thank you very much, Professor Anderson. So it's very fantastic and also wonderful lecture. So anyway, so we have some question here. So Dr. Noto, so would you like to say directly for the question?
If people are comfortable putting their videos on, it would be lovely to see everybody. But if you're not comfortable, don't worry about it. You can come in by typing or by speaking. Either is fine.
Thank you very much. So anyway, Dr. Farid here. So please, Dr. Farid. Yes.
Yeah, thank you, Professor Anderson. Very interesting lecture and very nice lectures. I have several questions. The first one is, I don't know whether, I think you haven't mentioned about the definition itself of caloric restriction in term, I don't know, because you used the monkey models, and I think you also showed the human model. In what, to what extent that you perform this caloric restriction in the experiment, like how many calories reduction and for how long, or the nutrition itself, like the type of nutrition, it also matters in this caloric restriction procedures or experiments.
Sure.
That's
okay,
my
so
first question.
Yeah, and I apologize. I should have mentioned that. So in the monkeys, we started out with the 30% restriction, but as the animals became old, the control animals ate less. So it shrank down to more like 25. And then by the time the animals were very old, we didn't really have good numbers, because we started with 76. And then they dwindled, of course, by 27 years of age, we were down to only half of the controls, for example. So we started out with 30% restriction in that. In the mouse data that I showed, that aging, that was an 18% restriction. So far less restriction that was able to actually produce survival differences, but also molecular signatures. And we've looked at a range of different tissues from those particular animals. In the human study, the target was 25. And I think what they actually got in aggregate was about 15. And so initially they started by going, oh, well, let's impute the data, imagining everyone did 25, which is a bad idea. I think imputing data is a bad idea. And more recently, they've started to be more granular in their analysis. And so they'll look at responders, people who were compliant and stuck to the diet versus those who didn't. And then they'll look at grades of compliance. So if somebody was on 20% CR, how do they differ from someone who only managed 7? Because the average compliance was 15. And I think this is something that we've seen in the clinical literature, being able to look in and see the data that you actually got instead of what you had hoped to get is a much cleverer way of trying to pull out the biology. So in human studies, I think it's safe to say people could definitely tolerate 10 or 15%. And I have no reason to believe that that would not confer benefits. Traditionally in mice, people went as high as 40% CR, 50% CR, or 45% I think was the highest ever done. They're extremely long-lived mice. No one on planet Earth that is a person is going to do that. I know, for example, for myself, if I don't have lunch, everything goes very strange by 4 p.m. So different people will be able to tolerate different things. And that's part of why many of my colleagues are more interested now in things like time-restricted feeding. So to your last point about the composition of the diet, I think what we need to realize is it matters how much you're eating crossed with what you are eating. So if you're on a CR diet, you can eat whatever the heck you like because you're using all of it, you're storing little of it. So, you could, your food could be things that are associated with poor health, but because it's all being used and it's all being metabolized, you don't have that negative consequence of adiposity and potential inflammatory or glucoregulatory dysfunction. If you're eating too much, that in itself is the problem. So if you're still eating too much, switching to healthy salad and still having 2,500 calories is not healthy. So you can't change the composition and hope that it would overcome overeating. But in the middle zone, which is the sweet spot, absolutely what you can eat can dictate health status. And so I would point you to a review that I just finished with my friend Walter Longo. We published it. It came out last month, but this month, in Cell, and it's really his baby, the longevity diet, and that shows the types of things that have been associated with health in human populations for which we have evidence from model organisms that those date changes are associated with longevity pathway regulation. So it's a nice paper. So it does matter what you eat in terms of composition, but it really matters how much you eat, and the rules will be different depending on that. Okay, that was a long answer. My apologies.
Thank you. Very interesting. And maybe my second question is regarding the molecular mimicry of the caloric restriction, because you mentioned the intervention regarding the mitochondrial. Everything is changed in the mitochondrial levels. So would it be possible, or I don't know, maybe the target itself in the future, you want to make this molecule that mimic the mitochondrial changes in this caloric restriction? So maybe a lot of people would not want to restrict their calories, but they just want to eat medication in certain. Is it, I don't know whether that's already being done, or
I can't tell you the millions and billions. Lots of very, very wealthy, now getting older people are interested in precisely that question. And so there are a number of drugs that are of interest. So something like metformin was followed a lot, and it seems to be effective, but only in correcting an existing metabolic dysfunction. You can't take an already healthy person and push them into greater health, but you can take an unhealthy person and bring them into, good health. So not everything that works at one level, like correcting a high-fat diet, for example, will then also work on longevity. One of the molecules that we're interested in is, sorry, there's something beeping out there. Where is it? If you can just, is it in here? Oh, it's in your jacket or something. Okay, thanks. I have a beeping backpack in the room with me. Where was I? Yes, we're interested in a drug that's called adipiron because it targets adiponectin receptors. And we think that that's going to be of value because adiponectin is one of those adipose-derived molecules that increases with CR. So we think it's going to be very aligned with the normal physiology of caloric restriction to go after this pathway. And so we've been looking at that as a means to preserve skeletal muscle. And I can tell you it works, but only in males. So, oh gosh, me and all the girls on the planet will have to find something else. The boys are okay though.
Great, thank you, Professor.
Lovely to meet you.
Yes,
you.
thank you.
Okay, so we have another question from the Dr. Noto. Maybe as you know also that the elderly population has like a undernutrition phenotype.
Yes.
So what do you think if we can translate your CR diet research into this undernutrition? Yeah.
This is a super important question. So thank you for posting it. So there's a difference. I think one of the things that we have to really grapple with is that recommendations that we make to young people are not the same recommendations we would make to middle-aged people, and certainly not what we would recommend to elderly people. So there even is evidence to this, where a low protein diet is thought to be beneficial, and that looks good in the clinical literature. And this is work from Walter Longo and Morgan Levine from a few years ago. But at 65, that relationship changes. So low protein is not indicated as a good idea in the over 65s. Now, this is population level studies. So we all know once population level studies are super interesting, they give us good clues, but they are not a means to make recommendations to individuals. Because what happens in population studies is things get smoothened out and we can see various trends. There's no room for nuance, there's no room for detail. And this idea of one size fits all we know is completely untrue, because actually one size fits nobody. One of the things we're acutely aware of, and I've seen this among my geriatrician colleagues, but also in the clinical literature, is that avoidance of food, especially in the very end stages of a person's life. And that is not caloric restriction, that is going in towards malnutrition. And it's one of the most difficult things with dysphagia, but also, problems with what I would call the plumbing, incontinence or constipation, these problems in the digestive tract and removal systems that people are off their food, they don't want to eat. And once you see that decline in food intake, it's a very precipitous decline in terms of other morbidities and eventual mortality. So that we haven't really nailed down the recommendations in older populations. My friends at NIA had a monkey study where they did later adult onset. So these were monkeys that began on the diet at 20 years of age. So this is still ahead of the 50% mortality that happens around 27 years of age. So this would be like in their 50s people, maybe. And they were able to show benefits there. But I think the real benefit of caloric restriction work is to uncover the biology. And I've always stopped short in terms of trying to translate the intervention. Rather, I'd like to see the translation of the mechanism. So I think it's a very important point, Noto, that was brought up there, that how we think in terms of clinical treatment of older care, we have to be careful not to go nuts with the latest in science. It has to be tempered for older individuals. And, as I've learned myself in teaching the medical school classes, many of the people over 85, for example, are on 15 different medications, and they have a constellation of morbidities. So usually they come in and there's a question of, well, which one are we going to look at today? Which one is the most important? And so things like food and nutrition really go into a different realm in the very old individuals. I hope that answers your question.
Okay, thank you very much. So actually, I know that paper that published in Cell Metabolism, Levin and also the Walter de Longo. So actually, I would like to ask the following questions about that. Is that like specific nutrition matters? Like maybe as you mentioned that the high protein diet is beneficial in the more than 65 years old, maybe, or some research I saw that the BCAA deficient diet maybe is beneficial, and also. Yeah. So what do you think about that?
there are a lot of individuals looking at the branched chain amino acids, but I can tell you, the lion's share of what people are doing in dietary studies is against a so-called Western 40% fat diet, or sometimes the high fat diet, which is 60% fat, which is completely lipotoxic for a mouse. And so I think it becomes nuanced. I think using diet as a medicine is something that's very popular. People would like to be able to do it, but part of me is very practical, and maybe it's because I'm Irish. It's clear to me that by modulating how much you eat and staying active, and this is something that your grandparents could have told you is the way to stay healthy. Eat in moderation and stay active. The interesting thing about the diet stuff that I did with Bolter is when you look at the clinical literature and peel away the things that are to counteract overweight, obesity, and diabetes, because that's a separate condition, it becomes quite clear that, making changes like having more protein from plants than from animals is a good idea. It doesn't mean don't eat meat. It just means don't eat it at every meal. Also animal fat versus plant fat. And it doesn't mean eliminate all dairy. It just means go easy on it. And so there are these tricks, and you can see it from the longevity diet, mostly from plants, avoid from animals, and then having this balance in the diet. But we also know that different people will respond differently. We have differences depending on how old you are. Are you on medications? Are you male or female or something else? If you're a woman, are you postmenopausal or perimenopausal? So all of these factors will have an impact on how the diet is processed by your body and how it affects adiposity. And adiposity seems to be the main thing to really avoid. Although that said, we know there are people who have extra weight and are not unhealthy. So it's an interesting thing that for a lot of people, this is so confusing. Is it good to be fat or bad to be fat? And I think if you think about it, it's the function of your adipose that is important, not necessarily how much of it there is. So you can have somebody who has a lot of adiposity, but it's not inflamed. They don't have inflammation associated with that adiposity. So they're not going to be as unhealthy as someone, say, who's less fat, but is very inflamed and diabetic, for example. So the detail, it's all in the detail.
Yeah,
and I can see,
agree.
I'm just going to wave to Genti there, a former Madison resident. I have to say, go Badgers, right? That's what they say here.
Hi, doctor. Thank you. Nice talking and meeting you.
Nice to see you.
Thank you.
Thank you very much. So yeah, I agree with your previous statement that the currently published in the Nature Aging that somehow the obese population in China has a longer lifespan. So yeah, it's very interesting about that. And maybe the quality is more important than the quantity. Yeah.
This is, yes. And this is something that we've been looking at this in the context of caloric restriction, because everyone goes, oh, less adiposity. And it's really only in the last 15 or 20 years that it's become clear that adipose tissue is an endocrine organ. So I'm interested in the adipokines and lipokines, but as fat expands, you get infiltration of immune and inflammatory cells. So you see macrophages, eosinophils, and they're all in there. And then depending on how they behave, they will contribute to inflammatory tone. So there's this thing called inflammaging, which is a compound word. Gosh, I hate when people make things up, but this one has been very useful. So inflammaging is this idea that a low-grade inflammatory tone has contributed to disease vulnerability as a function of age. And it's not like the inflammation you would get if someone whacked you or you were recovering from a disease or blunt force trauma. It's a low-grade persistent. It's like raising the baseline. And then what happens then when you do have an injury or a challenge, you overshoot and then may fail to resolve. So this dysregulation of immune and inflammatory pathways that we know happens over the course of aging, of course, I think it's to do with growth signaling, and that's a contributing factor. So yeah, and I think it's going to depend then. So it's not just how much fat you have, it's how that's behaving. Is it inflamed? Is there evidence of dysregulation of glucose handling, insulin sensitivity? And all of these will rack up. But I'm sure many of you will know this working in the area of metabolic syndrome, where we consider a constellation of features, any 5 of the following 13. And so it's the same way that we approach aging and nutrition studies with the same level eye for detail.
Okay, thank you very much. So is there any question? So maybe while writing another question, so maybe the fasting question will be interesting also. So what do you think about the fasting compared the calorie restriction? Is that more superior, or maybe I mean some researchers mentioned that the calorie restriction is more susceptible into the infection disease, or so what do you think about the fasting research? So I'm because the fasting is more popular for us compared with the,
absolutely. And well, and you know, it's many religions had fasting in them. Some of them have cast it aside, but it was a component of many religions back in the day, and it's certainly healthful. So what I can tell you based on the studies, if we were all mice, the fasting is definitely health inducing. The animals that we and there's a number of people studying this time restricted feeding, or as we say in people, time restricted eating, and it is associated with benefits. You mentioned this thing about immune and inflammatory pathways, and I have to tell you, you know, this idea going through the literature that, oh, CR animals are bad at immune and infection, it's simply based on a couple of experiments done by an individual in the 90s, where they do the huge dose infection induction, and then the CR animals didn't do as well. But they didn't examine medium doses or low doses, and they didn't look for the trajectory of change. So if you can imagine in a growth depressed animal, it might take longer to launch a response, and it might look different in terms of, you know, the population of cells that are expanded, the order in which the, you know, immune cells, inflammatory cells, and then the cleanup crew come in for rejuvenation, for example. But unless you look all the way out, you can't say it's defective. It's different, not defective. And there was a paper in Cell Metabolism last year that showed that response to infection is not impaired in CR animals. So going back to the fasting, what's super interesting is you can see benefits in mice, that you'll see extended longevity and you'll see improved health. It's not in the same efficacy as caloric restriction, although caloric restriction, you must know, includes a fasting period. Because when we do caloric restriction, we give the animals food in the morning and then no more. They eat it all and then fast the rest of the time. So classic caloric restriction has a lower food intake and a fasting component to it, just by design. And but I can tell you in our monkeys, so all of those had food removed at 4 o'clock. So in that case, and they were eating the exact same diet, just less of it for the CR animals. In that case, we were looking at basically time restricted feeding versus time restricted feeding plus CR. And the animals did better with CR. So CR seems to have like this extra component that it introduces beyond just the fasting that is healthful. And like I say, look, I don't know who's really going to do CR. I think the value is going to be in learning what it does and why that makes a difference in terms of health and lifespan.
Yeah, so I saw that in the deadly lambing research together with you. So that's interesting combination.
He's in the Department of Medicine. We're in the same department. Yeah.
Oh, really?
We share. Yeah, I'm on mentoring committee for most of his students, so I see him all the time. Yeah.
Okay. So any other questions? So yeah, I'm sorry. It's maybe the time is very limited. So maybe one last question. So maybe it's kind of other focus. So I would like to know, how did you perform or maintain the monkey research for 30 years? So it's very long research. So how did you make it?
It's had it's had a number of people in a lead position. It started out with Joe Chemnitz in 1989. And then my former mentor, Rick Weindruck, who's very renowned in caloric restriction studies, took over in 1992 and expanded it. It started out with just a small group of males, 30 animals. Then another 46 animals were added in 1992. And it became a program project, which is a larger mechanism for studying. And it was maintained as a program project for several cycles. And then in 2010, there was a financial crisis and a very bad pay line. And so then only a few monkeys were left. Well, there were still monkeys left. We had to cancel the program project, stop doing any science, and only keep the monkeys alive on a smaller grant mechanism. And then the following cycle, we went for another smaller grant mechanism, but because there were fewer animals, we could bring the science back in. And now we have no animals left, and it's only the science. So we've gone from Rick leading it to then Ricky Coleman and Rick, and then me and Ricky Coleman, and now me. So, so we have it's like a musical chairs for leadership. But obviously, you know, Rick Weindruck was at the height of his career when he started it, and retired before it was finished. Joe Chemnitz is emeritus status. So these are the kind of things where you have to really petition for the money from NIH, NIA, and then have a leadership that really believes in it. But I don't think at its initial inception anyone thought they would be funded continuously for 30 years. They didn't know that when they started. So that's a good trick for anyone who's a junior investigator. It may end up lasting 30 years, so don't be afraid to begin.
Yeah, I hope so. Okay, so yeah, I think the time is already finished. So thank you very much, Professor Anderson, for your wonderful lecture. So I hope we could invite you again in our annual conference in the Eyes on Emery, maybe in the November, or maybe.
And I can't tell you how pleased I am to have had a chance to give you this talk and, well, meet everybody. And I'm so excited that you have this developing aging program. And if there's anything I can do to assist you, point you out to some good people to come and give talks at your seminar. And hopefully, if I meet any of you at an aging conference, please introduce yourselves, and I can help you to meet other people in the aging field. I'm so pleased for you that you're doing this. It's lovely. Terrific.
Okay. Thank you very much, Prof. Anderson. Okay, so
Bye.
see you. So I would like to finish this session. So thank you very much, Prof. Anderson.
Bye. Thank you all for coming in the evening. I appreciate you taking the time.
Okay,
bye-bye.
Bye-bye. Have a nice day. Thank you very much, Professor Anderson. Terima kasih, Yang Baik. Okay.